my name is Ed Fox and I work with CSIRO's Food Safety and Stability team. So we're based at Werribee and we do quite a lot of work with different food manufacturers in terms of both stability of food, so that's really dealing with spoilage, but also with safety of food. And quite a lot of that involves working with producers on their environmental monitoring program and just making sure that everything is working well. Okay, so I'll talk a little bit about 4 different sections: the tools and techniques that are involved in environmental sampling, verification of sanitizing or pathogen detection. So what's the question that's been asked? I'll talk a little bit about the zones and workflows, so how you can implement or bring zones and workflow segregation into an environmental monitoring plan. And then I'll briefly touch on the end on some other kind of more advanced techniques that we use for environmental programming. So Sally mentioned the case in the US where DNA fingerprinting was able to track something back, a contamination in years, so that had happened years previously. So I'll mention some of the types of techniques that we can use and we can actually implement into environmental monitoring to give us an even more robust system. So what are the tools we use for environmental monitoring and what are the technical approaches to this? There's a number of general considerations for environmental monitoring. So it may be pretty obvious you want to have as sterile a technique as possible, so you don't want to contaminate your sample yourself when the area you're sampling wasn't actually contaminated in the first place. And it's also very important that you use the correct sampling device to take the correct type of sample. And I'll give some examples of some of the different sampling devices that we use and when you might use which one. It's important that you sample appropriately, and then once those samples are taken, that they're transported to the lab where they're going to be tested, be it either a lab that's on site or a private testing lab, in a way that's going to give you the correct result and not change the result that you might get because it was mishandled. And also then it's important that the samples are tested in a timely manner. So you don't want to take a sample and then 5 days later go and actually test to see what your target bacteria was and if it's in there. So there's a number of different sampling devices that we use, and I'll show some of them at the little workshop later on when we're talking in a bit more detail about how to take a sample. But one of the main ones we use are sponge swabs. So these sponge swabs are basically dry sponges that are sterile in these bags, and then you can add some liquid to them and then use them to actually swab an area. So for example, if I was swabbing the surface of this table, I would typically use something like this, a sponge swab. What I wouldn't use is a stick swab. And often what you'll see is stick swabs are used quite a lot by people to take kind of large surface area samples. And that's not what they're for. So stick swabs are just a sort of a cotton bud, if you like, at the end of a stick. And what we use those for are, for example, if we were trying to sample a slicer or a piece of equipment where it's very difficult to get in to parts of it. Or, for example, often areas that can be problems for listeria are trolleys, or if you've got sort of open areas on the bottom of the trolley in the leg where listeria can actually get inside there, and that can potentially cross-contaminate the environment. So stick swabs are ideal for getting into those kind of more hard-to-reach areas. Another really useful swab that we use to take drain samples are what we call Moore swabs. So Moore swabs are essentially like a piece of cotton that's tied off, and you can actually put this in the drain, and we'll typically leave these in the drain for maybe 24 hours or even 48 hours. And it's a really good way to get sort of a snapshot of everything that went into that drain over a long period of time. So Sally mentioned that wet areas are key issues in terms of where listeria can colonize. And generally speaking, if you want to find listeria in a factory or a food processing facility, the best place to look is typically drains, because that's where you'll find them. And no matter wherever it is or whatever the process is, it will ultimately get washed into the drain at some stage. And it's in these drains that Listeria can colonize, and then from there can come back out of the drain and cross-contaminate the area. So more swabs are a really good way of actually seeing what's going into these drains over prolonged periods of time. And also then liquid samplers are we use for taking, for example, you might take a spot sample from a drain, or you might be taking a sample of milk from a bulk tank. So in that case, we would use these liquid samplers, which are effectively just little cups on the end of a stick, and then you can just take your sample and then cap it, and then take that sample for analysis. So with the sponge swabs and the stick swabs, which I guess are the real workhorses of environmental programming, how do we use them? Well, typically, as they're dry, if you're taking a sample, you will have to moisten them first. So usually what we do is we'll moisten them with some kind of a suitable liquid. Typically what we use is one called maximum recovery diluent, so you can buy this from suppliers. And this is essentially just a sort of a non-selective general media that will not have a negative impact on bacteria, because one of the issues of bacteria is so we say if Chronobacter is surviving in a dry environment, or Listeria is surviving in an environment that might not necessarily be very favorable, the bacteria survive, but they're not in the best of shape. So they can be quite sensitive to different stresses that can actually kill them, although if it contaminated a food, it may recover and then ultimately cause illness. So it's important that when you take the sample, you don't actually inadvertently kill the bacteria when you take the sample. So for that reason, we would never use a diluent that would actually affect the culturability of the bacteria, and in which case, there might have been Listeria in that sample, but you've killed it because you've used a media that has some kind of a selective agent in it that's going to kill the injured Listeria cells, for example, or the Chronobacter cells. And also, we don't use grout medium, because what can happen is, if you think about it, if you use a grout medium, so a grout medium is medium that bacteria can grow in, so it has all the nutrients that they need to grow. If you use that and then you take a sample, and then it takes you, we generally say you want to test the samples within 24 hours. So it may take you, within the space of a day to get the sample to the lab. The sample will actually change with that medium that so the bacteria in that sample some of them will grow, some of them mightn't, and it can potentially give you a different result. So it's important that you don't use a media that will support the growth of the bacteria when you take your sample. And one other quite useful solution that we use is called a neutralizing solution. So sometimes you want to sample an area where you know that there's residual disinfectant still there. And if the disinfectant is still there and you take your sample, it can still be working over time. And if it, as I said, if it takes a day to get there, within that day, the residual disinfectant may actually kill the Listeria that were there. So in that case, we would use a neutralizing solution, and this will just inactivate the disinfectant, and it means that it won't alter the actual sample that would lead to potentially a false negative result. For transport then of samples, it's important that we keep them at refrigeration temperatures. So Sally mentioned most of the bacteria that we test for, so Salmonella, Chronobacter, E. coli, they won't actually grow at refrigeration temperatures. So it's important that the sample is kept at refrigeration temperatures, and that doesn't affect the microflora. And again, Listeria will grow, although it will grow below 5 degrees, it will grow very, very slowly. So if you test it within 24 hours, that won't be an issue. It's also important that the samples don't get excessively frozen. So while Listeria itself, or Listeria monocytogenes, isn't quite as affected by freezing as some other organisms, the organisms like Chronobacter are. So if you freeze Chronobacter, it's probably going to kill it. So if you take a sample and then you freeze it, and there was Chronobacter in that sample, you're going to probably get a false negative result when you actually test it, because that actual freezing has a killing effect on organisms like Chronobacter or Salmonella. And it's important, again, that when you take the samples, they're protected from excess moisture getting in. So that can not only cross-contaminate the samples, but it can, again, affect the microflora in it. And generally, as I mentioned, the samples should reach the lab and be analysed within 24 hours of them being taken. Within 24 hours and being held at refrigeration temperatures, that's generally taken as a rule of thumb to be okay for testing that sample. So in terms of the tools and techniques we use, this is very much dictated by the target pathogen. So if you send a sample to a private testing lab, depending on what they're looking for, if they're looking for Listeria, they will have a standardized method that targets Listeria. So for example, there's specific growth medium that's used. I think we mentioned earlier, if you get a positive Listeria, it's usually on a Friday. Part of the reason for that is that often it can take up to 5 days to get the results. So if the sample goes in on Monday, you don't actually know that Listeria was in it until Friday. So, and for that reason, although we have specific Australian standard methods in place for how we detect Listeria in a sample, people will sometimes use other validated methods which are more rapid, so might have a turnaround time of 1 or 2 days, and will give you an indication if Listeria was in a sample or if Chronobacter was in that sample. So, and these standard methods generally have 2 different components. The first component is the detection. So it'll run through a method. So for example, in the case of Listeria, it's a double enrichment. So you take your sample, it's grown overnight in an enrichment media, it's transferred to a second growth medium, and then that's grown overnight again. And then, so you can see it takes a few days for this. And then when we essentially take that sample and put it on what we call an agar plate, which is just a selective plate that will kill other bacteria and prevent them from growing, but will allow Listeria to grow, so we can detect it in the sample. And then if there's a characteristic colony on that plate that is representative of Listeria, that's a presumptive positive. So it's not a confirmed positive yet, but the sample may have Listeria monocytogenes in it, or Chronobacter if that's what you're testing. And then there's a confirmation step to confirm whether it is Listeria monocytogenes or whether it's something else. So in the case of Listeria, when you get a presumptive positive, often you won't necessarily know what species of Listeria is, so it might be monocytogenes or it might actually just be a non-pathogenic Listeria. So there's a confirmation step that comes after that to identify that it's Listeria monocytogenes or it's Listeria innocua, or it's whatever organism you're testing. So the standard methods, as I mentioned, take quite a long time. So realistically speaking, for short shelf life foods, testing a sample and then having to wait 5 to 7 days for a result can be a problem. So often you might need to implement some more rapid methods just to get a grip of whether there may or may not have been contamination. And these rapid methods are quite useful to implement into an environmental monitoring program just through general production to get a feel for whether there may or may not have been a contamination event. And there's a number of these rapid methods that are approved by some kind of a standard, an organization that approves standard methods. The AOAC is one example, that's the Association of Analytical Communities, but there are a number of others. And if you, I won't go, I mean, there's a really large list of a lot of different methods that you can use for rapid detection of Listeria. Some of the really, I guess, easy ones to interpret are typically where you would take a cotton swab, take your sample, and then you put it back into a growth medium, and the medium turns black if Listeria monocytogenes is in there. So those are really useful, easy to interpret, typically only take about 24 hours, and they can be quite useful to do. For example, at the end of production, you can quickly do it on the line and get an idea for whether maybe there may have been a contamination for that production run. If this is the Food Safety and Inspection Service in the US has put together a really good list of a whole range of different rapid test methods, not just for Listeria, but also for Salmonella and other bacteria as well. So if you go to the FSIS website there, you'll be able to look, and you can actually download a spreadsheet which has all these different methods, and then potentially pick one that might be suitable for your application.
Excuse me, and that can be obtained by any specific operator, or don't have to go to a lab?
No, that's freely available to anybody online. So if you just go onto that website, or so if you go onto that website and search, just search it for foodborne pathogen test kits, it will take you to the page on their website that has, you can download like a PDF spreadsheet, or they've also got one in Excel, in Microsoft Excel, so you can download that as well, and then you can actually go through all the different methods, and it will give you some information on how they work, what they're suitable for, what organisms they work with, and that kind of thing. So it's a good resource if you're looking to select kind of, you know, a specific rapid test for what you want to test for. So for example, you might want, if you're testing a processing surface or a food contact surface, you might use something like the color change one on the swab, because that's quite rapid and is very easy to interpret. Some of them might require a piece of equipment and others don't, but certainly ones with color change don't tend to require, you know, you to buy some kind of instrument to actually analyze the result. So when we talk about actual environmental monitoring, then there's two reasons or two aspects to it. One is whether we're trying to verify that our sanitization is working. So Sally's touched on both of these in her talk. So just to make sure that we're actually cleaning and sanitizing and that we're not leaving Listeria or Chronobacter there afterwards. So making sure that that's working. And then the other one is that we're actually just doing more kind of surveillance, if you like. So just looking at the occurrence of different pathogens in the environment. And that's particularly important, as Sally mentioned, where there's a risk of post-processing contamination. So typically with a lot of these foods, there'll be like a cook step, for example, or pasteurization, which will kill Listeria, it will kill Chronobacter, it will kill Salmonella. But it's really the critical contamination point is after that cook step, where the bacteria in the environment make their way onto the actual food. And then ultimately into a human if they consume that contaminated food. So there's various different, I guess, sampling points, if you like, that can be that can be brought into an environmental monitoring program. Obviously, testing of food products is an important one, but as we know, when we test a food product, it tends to be what we call destructive sampling. So destructive sampling is where when you take the sample, that's it, the sample is gone. So if you take a piece of food and sample it, nobody can eat it after that, so it's gone. So as Sally mentioned, we obviously, we can't take every single piece of food that's produced and look for Listeria, or there's no food left to eat. So that's why we can do some sampling of our food products just to get an idea of if there has been a contamination during that production. But really it's the testing of the environmental sample is where we're really understanding what the potential risk is of post-processing contamination of these foods. Because if you've got a control step, like cooking to inactivate your target bacteria or pathogen, and you've got control of it in the environment and it's not in the environment, then it's not going to get a chance to contaminate your food. So when we talk about these environmental samples that we take around the facility, we can use these samples to test, so we can test food contact surfaces or non-contact surfaces, and then this can identify if there's an area that has contamination, but also if all of a sudden you get a lot of positives in a certain area, it may be indicative that there was some kind of a contamination event. So maybe someone walked it in, or there was Listeria resident in a drain and it got spread into the environment. So an example of that would be if sometimes what you'll see is hoses being used. So if you hose a drain and create an aerosol, the Listeria can stay in the air for hours. So it's not even just minutes, it's hours in the atmosphere, so you can in the air. So you can imagine that then has implications to move around and contaminate lots of other areas of the plant. Testing of ingredients is also useful, just to get an idea of the burden that you're introducing in your ingredients, or if there's a problem kind of further back the supply chain. So, I mean, the thing to remember with Listeria particularly is Listeria is what we say ubiquitous. So you find Listeria, Listeria is a soil bacteria that just happens to cause disease in humans. But I mean, its natural environment is to live in the soil. So obviously with agricultural systems or food production systems, you've got, you're practicing part of the food chain in an area that's also shared by Listeria. So there's lots of different ways for Listeria just to kind of coincidentally get into the food chain through production. So testing of ingredients is a good way to understand if be it milk coming in, has the milk got constant problems of Listeria contamination? And also then if it has, that will give you more of an appreciation of how to handle it when it comes in. So for example, if milk is coming in and it's been loaded and it's spread on the floor and there's Listeria in it and someone walks through it, they're going to walk it into the plant on the soles of their feet. So it's important. So ingredients can be quite valuable about getting an understanding of that. So in terms of when we sample, so we can sample during production or post-production. So this is really to understand what's happening as the food is being produced. So as Sally mentioned, as everything is going, your slicers are going, for example, if there's slicers or whatever, that's a good time to sample and understand if Listeria or Chronobacter, if it's that's your target organism, is being introduced into the process. So typically it's good to do this during production, but also it can be quite easier to do it after production, so post-production, before the sanitization happens. And the different food contact surfaces that we typically look at are the production line. So the production line to give us an indication if we get a positive detection of the bacteria in the production line, there's a good chance that it may have actually contaminated the food that moved along that production line. But also equipment, particularly equipment that has very hard to reach areas. So slicers are notoriously implicated in spreading Listeria or contaminating food with Listeria because it's so difficult to actually sanitize and clean some of this equipment. So typically what you'll see is at the end of the day's production, these slices will basically be taken apart and each of the blades and everything sanitized separately because just trying to sanitize it while it's intact can be quite difficult. So it's very important that if you have a slicer and you're sampling it afterwards and you're getting positives, well then the way it's been sanitized isn't necessarily working and you need to rethink how that's actually going to be done. In terms of non-food contact surfaces, then certainly what we've heard earlier on is wet areas are typically where you'll find Listeria, so particularly drains. In the case of Chronobacter, it tends to be dry areas, so we know unlike a lot of other bacteria, Chronobacter and Salmonella are really good at surviving in dry environments, much better than Listeria is. So this is potentially an issue, for example, if you've got a canning and a bag or a bag filling area. So those are really high risk areas in infant formula manufacture where Chronobacter can cross-contaminate. And again, in terms of ingredients, depending on your infant formula production chain, but wet processing tends to be a bit easier to control. But if there's some kind of dry blending involved, so be it vitamin or some kind of supplements into it, then obviously it's important to make sure that you're testing for Chronobacter in those ingredients that are going to go into the product as well, because typically there might not be a control step after it. And footwear is a really, really important vector that Listeria is able to utilize to move through the factory. So if you've got a drain and there's some kind of a splashback out of the drain, for example, or there's a wet area around it, just walking over that drain, and we've seen this so many times in factories, it typically moves the Listeria through the factory. So it's really important that, as Sally mentioned, everybody in the factory is aware of why we're doing this environmental, but also what are the key vectors, if you like, or risks for spreading Listeria. I was working in a large meat factory back in Ireland doing some environmental monitoring of Listeria, and I was trying to take drain samples, and there was a really good culture of understanding the risk of Listeria through this factory, and I would have to wait till everybody left the room so I could go and lift a drain to take a sample, or they'd just throw me out. So that kind of culture is really healthy, and it really reinforces because if people know why they're doing it, as Sally said, then they're likely to actually practice it. But if they don't know why they shouldn't be lifting a drain, then they'll potentially do it one time just for whatever reason they think it's necessary to do. One other aspect that we tend to implement quite a lot now in our environmental plans is making sure that obviously the cleaning and the sanitizing is working. So one example I have here is this was a sanitizer that was used to wash crates in a factory. And it was used so essentially where you see each of these different sets of bars was a different Listeria organism that we had isolated from that factory. And where you can see a bar, so you can see there's 4 different colors here, each of them can correspond to a different concentration of the sanitizer. It was used at 1.6% in this factory. So the crate wash was using this sanitizer at 1.6%. And what you can see, which is this sort of light bluey bar, and what you can see is every single one of the Listeria strains tested were resistant to this concentration, so it didn't kill them. And in fact, if we added a small little bit of nutrient in with the sanitizer, it would actually be able to grow in the presence of that sanitizer. So it's important to understand that the sanitizer you're using is actually going to inactivate Listeria. And I guess one other thing worth mentioning there is if Listeria is in a drain and sanitizer goes into a drain, straight away you've got a kind of a dilution effect because the sanitizer is going into more liquid. So it can also be that, you know, it might work at 1.6%, but it might not work at 0.8%. So it can be quite good to get an understanding of, even if you've got that dilution effect, that your sanitizer is still effective.
Can I just ask, that sanitizer, what type of sanitizer was it?
This one, I believe, was an alkaline-based sanitizer. But I guess the trick is it can be difficult to just look at a label and say this is definitely fine or that's definitely not fine, because we tested a number of alkaline sanitizers that might have had different concentrations of some of those alkaline compounds in them, and this one didn't work, and there was other ones there that were perfect. So this was quite a large factory and I think we tested about 10 different sanitizers, and most of them were excellent. All of them, I think, except for this one, were excellent. But again, it just meant that when the crates product had gone into the crates and then taken out of the crates to be packaged, and then the crates were brought back. So if there was Listeria got into that crate, it was constantly recontaminating everything else that went into it. So that just was one quick change that they were able to make to really, close off on that potential avenue for cross-contamination. This crate didn't. No. This crate didn't. But some of them might typically be used at 60-degree water, for example, or that. But in this case it wasn't. So it was just at the end of the line, and then the crates just got a rinse with it, and then they were brought back. Yeah. So depending on the size of the production setup, zones and workflows can be really helpful in trying to minimize the cross-contamination of different areas with Listeria or Chronobacter. So in terms of what type of zoning, well, that will depend on the product. So for example, if it's a meat product or a dairy product, typically what you'll see is lower high risk. So you might have low risk where the ingredients, for example, come in and there's some pre-processing. Then you'll have typically as soon as you pass the control step, so be it a cook step or a pasteurization step, you're into high care, and that's segregated. And then from high care, usually what you'll see is high care goes back into low care when you get to sort of after the product is packed and it's going into the whatever lorries to go somewhere else. That will generally go back to low care. Or in the case of infant formula, the zoning tends to be wet zones and dry zones. So initially you have your wet zones, but as soon as you get towards spray drying, canning, filling, that will be a dry zone. So use of moisture is minimized in the dry zone. And then from there it goes. The product obviously goes out then to market. So this is just a kind of an example here, if you had of a crude example of where you might have ingredients, for example, coming in and some kind of pre-processing in green here, and this would be a low risk zone. And often what you'll have is you'll have as the product moves into, so for example, the product goes through a pasteurizer and then it comes out in the high risk zone, or the product goes through a cooker and comes out in the high risk zone. And often what you'll have is segregation of even staff. So you'll have staff that work on the low care side, staff that work on the high care side, and that keeps that level of segregation. Because the thing to remember, particularly with a lot of these bacteria like Listeria is in so many different environments, it's really difficult to keep it out. So this is the case for every factory. There's so many opportunities for Listeria to get in. It happens everybody. So it really is important that you kind of have a good control of it, so that if that does happen and the Listeria gets in on the low care side, it's not getting into the high care side. And some of the issues we've had with this is even with, for example, Sally mentioned some factories, if you like, are just not necessarily suitable for production. So sometimes if you're trying to retrofit something that was used for, mightn't have even been used for food production, to make it into food production, we had one processing factory I was working in where the drains flowed from low risk into high risk. So that was a massive problem straight away, because Listeria ends up in the drain. It's washed into the drain and then it flows through to high care, and then from there it was able to start to contaminate out into the environment. In another factory I was working in, we had a whole load of positives in one area, high care, and we came in and we were looking to see what was going on, and it was at one of the interfaces. So typically you'll have barriers between high care and low care. So that may be a barrier that limits access of staff, or also an interface. So if you imagine, for example, if product goes through a cooker and comes into a high care, and then it moves along the production, and then it will move out into low care where it's packed. And so there's obviously some kind of a window, if you like, typically where the product will move off the line and into high care. So that's what we call an interface, or a vulnerable point. And in the case of this factory where we were getting all the positives, what was happening was trolleys were going into the cooker on low care. So the door would be closed on low care, product would be cooked, product came out the other side into high care. The product was taken off then to go onto the actual line to be packaged. And then the trolleys moved into an annex room. So a door would open, the trolley would go in, the door would close, the door would open on the other side, the trolley would be taken out back into low care to continue the cycle. But just outside that door was the trolley wash. So there was a guy standing there with a hose, and he was blasting every hose. So for the entire production, this trolley was being, this was just so there was moisture everywhere. So what was happening was, all the Listeria that was getting into the moisture in there, every time they opened the door, some of it would move in, and then they'd open the door on the other side and it was straight into high care. And as I mentioned before, it can be, Listeria will stay in the air for a long time after you aerosolize it. So hoses in drains are a big no-no with Listeria. Sally covered off on most of this in her presentation. So it's important that staff know what they're doing. So for example, and that they have their responsibilities, and that they don't necessarily kind of do things. So the example I would have is, if you have staff working on a production line and some product falls off onto the ground, they're not going down to handle it and put it in the bin and then come back and do the production line. So there's cleaning. So typically, for example, what you might see is cleaning staff that come in and they take care of all of that. And never the twain shall meet in terms of production line staff looking after that side of things, just to minimize the chance that you're going to take Listeria from the environment and bring it onto the production line. And also, in terms of workflows, then footfall can be really important. So it may be that there's an area that's more prone to sporadic contamination by the organism that you're testing for. So be it Listeria at a drain or Chronobacter near a spray dryer. So sometimes it can be really useful even to just limit footfall in that area. So just keep it as barriered off so that people just don't necessarily walk through it in their general workflow. And obviously the movement of trolleys is a big one for spreading Listeria around, because if you move if you're pushing a trolley around the ground and it runs through a puddle that has Listeria in it, it's going to spread it around the plant.
Excuse me, just from experience, dishwashers again is human washings for equipment or in the premises. What do you think?
Dishwashers.
Just a normal dishwasher for any hardware you use for cleaning contents.
I mean, if you're running it through a dishwasher with a high temperature, again, a lot of these bacteria don't necessarily like the high temperature, but I would say you would still need to be conscious of not getting, because I mean you're going to be introducing moisture now, so it's just really important that that's controlled and there's not moisture being spread around the place. So, like, I mean, if you've got a puddle or condensation building up on the side, I mean that's a prime spot for Listeria to get into and persist there for a long time. And we're understanding more and more now that some different strains of Listeria seem to be able to persist for very long time. So we had one small cheese factory in Ireland that we isolated Listeria for. There had been some samples taken 10 years before with some other work that was done in this factory, and it just happened that the isolates were still in a freezer. And we then had a project where we were just coming in to kind of look at the environmental monitoring program and then fix up any suggestions that we could to improve it. And so we got some Listeria isolates and then we fingerprinted them, and sure enough, the isolates that they'd found 10 years previously were the same. So that same Listeria strain had stayed in that factory for over 10 years. So some of these bacteria, some of these persistent strains can survive for a long time. And now we're doing quite a lot of analysis of the genetic background, the genetic traits of these bacteria. And we know that some of them have, for example, mechanisms where that give them resistance to, increased resistance to certain sanitizers. Now, generally speaking, if your sanitizer is effective against Listeria, it will still be effective against some of these more resistant ones. But where it can start to come into play is, as I mentioned, you've got like dilution effects. So for example, if you've got a sanitizer, it may kill the Listeria if it's on the surface here dry, but if it's in a drain and the sanitizer gets washed down the drain, that sanitizer will be diluted and it might not kill the Listeria. And then that's further compounded by what Sally talked about earlier on, by biofilms. So what happens with biofilms is they form like it's like a mucus layer, if you like. And slime, exactly. And once the bacteria are in there, they're protected from what's on the outside. And that's why, as Sally mentioned, cleaning is so critical. Because if the cleaning doesn't work, the sanitization probably won't work. So that's an important consideration as well. In terms of the sampling plan and where would you sample, that again will depend on what the target bacteria is. So if it's Listeria, you definitely want to be looking in drains. You want to be looking in areas where you've got wet, kind of damp areas, or areas that are prone to continual condensation, for example, and also kind of hard to reach areas. So whether that's a slicer or whether that's, as I mentioned, even inside the leg of a trolley. So Listeria can get into these niches, form a biofilm, and just constantly just shed off the biofilm and recontaminate the area. With Chronobacter, you would want to be targeting the dry areas or the vulnerable areas. So for example, where you've got your canning or your filling processing, that's an area where you would really need to keep on top of any Chronobacter that might be there. In terms of how often, again, as Sally mentioned, that will kind of depend on the process itself. In terms of the actual the sample point, if it's a food contact surface, you probably want to do that at higher frequency than some of the environmental, because ultimately, if, for example, if it's on a processing line, that's really where it's got the maximum chance of getting into your product. So you would target those at a higher frequency than, for example, if you were taking just a non-contact environmental sample. And also, the sampling frequency should definitely take into account responsive actions. So if you get a positive sample in a drain, you probably want to home in and increase the frequency of samples that that are on that drain after you've taken your corrective action, to be sure that you've got the listeria, for example, if it's listeria in a drain, that it's gone. And then you would go back down to your normal sampling frequency. Another time where you'll frequently get increases in positive detections of bacteria of some of these pathogens is if there's some kind of retrofitting going on or some kind of maintenance works or building works. So typically, if there's a certain part of the plant, for example, that's been refitted, you would increase the sampling area around that just to make sure that nothing's been, that increased risk there isn't going to start introducing bacteria or listeria or chronobacter back into your actual production area. So when we're trying to decide on what we want to test for, Chronobacter and Salmonella are very important in infant formula. Listeria, very important in ready-to-eat foods or dairy products. But other organisms that are typically tested for are what we call indicator organisms. So an indicator organism is an organism that indicates that something has happened. So, for example, in the case of coliforms, that's typically used in water testing to know if there's been fecal contamination of water. So those are called indicator organisms, and they can be quite useful for understanding if there's been a breakdown somewhere in the chain in the safety or a contamination event, or also if there's inadequate sanitizing. So if you're pulling these bacteria off, it probably means that they're not being killed by the sanitization, and that can be a problem. And then index organisms, Sally mentioned already, are typically, for example, in the case of Listeria monocytogenes, the index organisms are other species of Listeria that will thrive in the same environments that Listeria monocytogenes does. So when we talk about Listeria monocytogenes is the one that causes infection in humans. And that's the one that we're really concerned of from our public health perspective. But there's a number of other Listeria species which are not pathogenic to humans, but live in the exact same niches as Listeria monocytogenes. So it's likely that if you've got a persistent Listeria innocua, which is not pathogenic, that's an indication. If you're constantly finding Listeria innocua in the same drain or the same environment, and you think, oh, well, it's okay because it's not Listeria monocytogenes, the implications of that are that if Listeria monocytogenes does get in there, it will do the exact same thing. It will persist there, and then it will potentially recontaminate the other areas of the plant. So index organisms like that are very useful to get a grip for whether if Listeria monocytogenes, for example, did get in there, would it be able to actually colonize? And the last thing I just want to touch on now is some of the what we call more advanced technologies or tools that we can implement into our environmental monitoring program to just give us extra information on what's going on. So Sally mentioned fingerprinting, and I'll just give you an example of how we use fingerprinting as part of an environmental monitoring plan. So here's our crude facility again, where we've got some positive, so each of those red spots is we're after getting a positive Listeria monocytogenes. So we've got a positive sample, so a couple of our ingredients that came in were positive. We've got a positive in the processing area, a positive, for example, in somewhere that's been further processed, be it a ripening room, and then a positive in the packaging. So we don't know how those are linked. We just know that there was 5 Listeria monocytogenes detected in different parts of the process. So one of the techniques we used, the fingerprinting techniques, is called pulse field gel electrophoresis. So this is essentially, if you know in forensics or CSI, they take a DNA sample of someone and say, that's the killer. So we can do the same, we can do the exact same thing with bacteria, where we can essentially take a fingerprint of the bacteria, and then we can say, that's this strain and this is a completely different strain, or that's the same strain. So that's the same strain that we found in 2 different parts of the plant. And essentially what we do is we get what we call a DNA fingerprint or a DNA profile. So each of these 3 profiles are DNA profiles. And you'll notice each of those little lines is a different DNA band. And you'll see that the 3 of them are different. So we can look at those and say, right, these are different DNA profiles. So those are 3 different strains of Listeria monocytogenes. And in public health, if there's an outbreak, for example, this is how they figure out where the outbreak came from. So they take the isolate from the patient, they get the DNA fingerprint from it, and then they go back to, they say, what foods did you eat in the last, with Listeria it's quite complicated because it can take 2 months, but they'll say, what foods did you eat? They'll go back and so if 10 people are sick and 10 people say they ate brand X of a meat or brand Y of a cheese, they'll go back to that product, test it. Any Listeria monocytogenes they find, they'll take a DNA fingerprint, and then as soon as they find the match, so they know the fingerprint that was in the patient is the same as the one in the food, that's how they link the foods to the outbreak. But
Unfortunately, the question goes, did you eat this? Not what did you eat?
well, yeah, exactly. Yeah, and I mean, there's a whole level of nuances in terms of how these epidemiological investigations and links are done, but crudely speaking, that kind of describes it. So if we go back to our example, so if we say each of these 3 different profiles, so we've taken our 5 isolates, and these are the 3 different profiles we found with our isolates. So each of them is now marked with a different colour. So you've got the red one, the blue one, and the yellow one. So what you can see here is this yellow one, so this Listeria strain that we found in the ripening room was the same strain that we found in the packaging room. So it's quite likely that we've taken the Listeria from the ripening room and brought it into the packaging room when we brought that product out of the ripening room. Or vice versa, if we had, for example, here's the positive that we had in the milk ingredient, and now we can see that it's in the processing area. So maybe that it was spilt and it was walked into the factory, or it was brought in some way into the factory. So the benefit of this type of approach is that we can see, first of all, we can see if there's been a contamination event and how it's after contaminating other areas of the plant. But it also, as we do this over time, we can see, after 1 year, 2 years, is the same bacteria constantly popping up in the environment? And if it is, it may be that it's got some kind of a niche in that environment and it's not being eradicated from that niche, and then it's periodically cross-contaminating. So this is really a really powerful way to home in on where the Listeria has colonized in the factory, and then get rid of it from that niche and make sure that it's not going to subsequently cross-contaminate later on. So to finish up then, I guess some of the important points with an environmental monitoring plan is use the right tool for the job. So don't use a cotton bud swab to try and swab the entire surface of this table, because it's not the right way to do it. So we've got specific sampling devices that are used to take specific samples, and it's important that those are used correctly and that the right technique of sampling is used. Environmental testing is really critical to achieving products free of pathogens. So we can't test every piece of food. So the best way to minimize the chance of Listeria or Chronobacter getting into our food product is by conducting environmental monitoring and understanding what's actually present in what the background level in the environment is, and what's that risk then for cross-contamination of our product. Controlled process and workflows are really helpful. Zoning can be really beneficial if you prevent crossover from the kind of more high, from the low risk area into the high risk area. And it's really important that everybody in all parts of the process and every person involved in it has an understanding of why food safety culture is important, and why they should do what they do, and what the danger, if there's a breakdown, for example, and Listeria can contaminate a food, is. And also then, some of these more advanced tools, such as pulsed field gel electrophoresis, can give you that extra level of information on what the dynamics of the cross-contamination are in the factory, and then allow you to home in on where potential breakdowns or problem areas are. So those are my contact details. I think they're in the slide. So if you have any questions on any aspects of that, just feel free to give me an email or give me a phone call and I can try and answer them.